The overall goal of this procedure is to amplify a specific DNA sequence using the PCR technique, to analyze the features of the primers, visualize and analyze the quality of the amplified product on an agar gel.
The most revolutionary technique in the genetic engineering field is the polymerase chain reaction, or PCR, the in vitro production of large quantities of a concrete and specific DNA sequence without appealing to a host organism's clonation by means of primers, forward and reverse, in a DNA polymerase of high and low fidelity. PCR consists of three stages for amplifying a sequence. The first one is DNA denaturing to obtain a single-stranded DNA molecule. The second one, alignment, so that the primer can anneal two complementary sequences. The third one is chain elongation for enzymatic activity.
Furthermore, PCR has many variants with different procedures and applications. In order to choose a PCR technique, it should be considered the needs of the research. Some of them are the nested PCR that increases sensitivity, the RT-PCR to obtain cDNA, the qRT-PCR to quantify, the PCR multiplex which amplifies different DNA sequences simultaneously, and the hot start PCR offers inactivated polymerases at room temperature.
What is required for performing PCR? DNTPs, primers, DNA template, DNA polymerase, buffer, trace chloride magnesium, primer design. Consider the following situation: amplifying P53. Gene, it's a tumor protein P53 in Homo sapiens. Check out this gene sequence at NCBI by the following reference number. Once it is found the coding sequence of the gene P53, the DNA template to be amplified, it is important to obtain the reverse complementarity of the original sequence in order to design the reverse primer. Both primers should be in a direction 5' to 3'. The reverse complementary can be obtained from the following link. Designing the forward and reverse primers with the help of the following link. However, for the primer design, it must be considered size between 18 to 30 nucleotides, melting temperatures between 50 to 65 Celsius degrees, and guanines and cytosines percentage content between 40 to 60. Self-complementarity, it should be avoided in case of herb information and primer dimers. It's recommended to establish an annealing temperature of -5 Celsius degrees with respect to the melting temperature.
An example of an application of a PCR would be the sexing of human DNA. There was the usage of a specific primers regarding ZFX and ZFY, genes coding for zinc fingers protein. However, in order to determine sex, homologous introns from the X and Y gene sexual version of ZFX, ZFY are amplified with a specific primer pair. Then, an alu element insertion of the ZFX, which is a restriction site for that specific intron, results in a size difference in the amplicons, which is visualized by an electrophoresis gel.
A master mix was prepared to deliver 47 microliters to 3 tubes, each one for the sample, the positive and the negative control under the conditions shown on screen.
Once the 3 tubes were obtained, to the tube A, 3 microliters of genomic DNA were added. To the tube B, 3 microliters of water for the negative control, and to the tube C, 2 microliters of water and 1 of PCR product.
The thermocycler was set with the conditions shown on screen.
For analyzing the PCR done, an electrophoresis gel of agarose at 8% was run for 40 minutes at 120 volts. 3 microliters of intercalating agent gel rate 0.3x were added in the agarose TBE solution. Tie running buffer was used. 4 microliters of loading buffer were used for each sample. In the first well, 6 microliters of 1 kilobase oxygen DNA ladder molecular weight marker were added, followed by the sample, the negative and the positive controls in wells 2, 3, and 4 respectively.
In the first well, it can be observed the DNA ladder, and it signalizes the size of the PCR product of the template. In the second well, briefly minor to 500 base pairs. A PCR product of the same size was noted in the fourth well, the positive control, but with the difference that this last one resulted in a thicker band and degraded. No PCR product of the size mentioned was identified in the third well, the negative control. In wells 2, 3, and 4, there were noted 2 bands so small that its size cannot be said. They go further the DNA ladder.
Regarding TNTPs, protection and stability, all nucleotides must be diluted in water, and then this solution ought to be protected by adding trace buffer 10 millimolar at a pH 7.7 to 8. This is due to avoiding acidic pH that could cause DNTPs hydrolysis, and thus they will not be able to be used for polymerization reactions carried out by DNA polymerase. DNTP's stability during repeated PCR cycles is high, so that 50% of DNTPs will remain as DNTPs for more than 50 cycles. Reaching DNA polymerase specificity and fidelity can be encouraged by means of preparing equimolar concentrations for minimizing errors when nucleotides are incorporated by DNA polymerase, and include the lowest adequate concentration of DNTPs for reaching length and composition of the target sequence. Oligonucleotides must contain between 40 to 60 GC percentage. Likewise, it is not recommendable to have large ATs and GCs regions because there could be a weak hybridization between template and primers and could cause secondary structures formation. Besides, it is important to have a primer length between 18 to 30 nucleotides, as it is considered as an optimal length. Small length primers could bring out unspecific PCR products, and long length primers could decrease efficiency when joining to the template. In this case, for the ZFX, ZFY amplification, a forward and reverse primer were considered respectively. It can be known from the forward primer 44 GCs percentage content, and from the reverse primer a 40 GCs% content. Thus, both primers have an acceptable GCs content and a length of 25 nucleotides.
It is highly recommendable to have a pure and integral DNA or RNA template that is free of contaminants such as phenol, proteins, and salts. For genomic DNA, it should be used a quantity between 10 to 250 nanograms for an adequate PCR reaction. In this case, it is proposed to use 150 nanograms of genomic DNA. It's important to mention that the DNA needs to be dissolved in pure water or in a trace from 5 to 10 millimolar solution at a pH between 7 to 8, avoiding EDTA that could kidnap magnesium ions needed for the DNA's polymerase enzymatic activity. In order to carry out a PCR reaction, it is relevant to make a master mix composed of trace, buffer, DNTPs, forward and reverse primers, Taq polymerase, DNA template, and water, so that a positive and negative control could enhance results when verifying a specific DNA fragment amplification. Regarding DNA fragment quantity for carrying out a PCR reaction, a total amount of 150 nanograms were selected as the calculation basis, taking into account a range between 10 to 250 nanograms. Then, a total concentration of 50 nanograms per microliter was selected as the desired concentration by the genetic engineers laboratory teams. Thus, every PCR microcentrifuge tube will have 3 microliters of DNA template. These standard values were chosen due to multiplicity in DNA template concentration among the team groups. An homogeneous concentration among the group will be a manner of verifying success in DNA amplification. For the positive control, we use less amount of template due to the standard values that corresponds to PCR 0.1 to 20 nanograms.
According to the 1 kilobase plus oxygen DNA ladder, there was an efficient amplification of the gene corresponding to 447 base pairs, as well as an equal value with respect to fourth well, the positive control that had the same size. Meanwhile, on the second well, a negative control demonstrated lack of contamination and lack of PCR reaction. Thus, adequate pipetting techniques and protocol management were carried out. The results obtained revealed that it was successfully amplified the gene due to the size observed that corresponds to the already known value. The fourth well presented the PCR product, but it resulted in a thicker band. Regarding the 2 small bands observed, its size cannot be said because they go further the DNA ladder. They may correspond to the primers, but both primers have the same size of 25 base pairs, probably the smallest band. This suggests that the other bands shall be in specific DNA amplified fragments. Due to the absence of DNA template in the negative control, it was concluded that those probable in specific amplified fragments could come from the primer's complementarity, and it was confirmed with the primer blast.
Nowadays, molecular diagnosis through gene detection and mutant variants, sequenciation methodologies, and gene amplification for a later cloning expressing recombinant proteins belong to the multiple PCR applications that take advantage of an innate biological process such as DNA replication. Nevertheless, an adequate nucleic acid extraction, verified purity and quality by a spectrophotometer and a gel electrophoresis, as well as an adequate management of reagents such as DNTP's protection and stability in a trace buffer, primer features, taking into account length, GCs, percentage content, melting and analytic temperatures, and avoiding secondary structures and primer dimers, and DNA RNA template amounts. If it is genomic DNA, use quantities between 10 to 250 nanograms, and if it is plasmidic DNA, use quantities between 0.1 to 15 nanograms.
There is an important checkpoint regarding PCR efficiency, such as a performing PCR reaction derived from a master mix in 3 different microcentrifuge tubes, representing the DNA target sequence, a negative control and a positive control.
Finally, PCR has multiple variants, and thus the researchers as well as the scientists can apply them depending on their needs. For example, if there is the need to improve a molecular diagnosis of sequences, a precise and effective diagnosis can be done. Microbial pathogens, specific DNA sequences coding for the specific proteins from these pathogens should be known, so by means of performing a PCR multiplex after having made specific primers for this.